Hello everyone, we're starting the last part 4 of Blood Flow 2 right now. We want to talk about the control of the resistance in arterioles. In other words, that vasoconstriction and the vasodilation is coming from where? Where is that control happening? So it's controlled both by systemic control mechanism or a local control mechanism. Those are going to be quite different. So when it's local, you have vascular smooth muscle, right? The smooth muscle of that region can regulate its own contraction. And this is pretty cool that it can do it by itself on its own. And that's called myogenic autoregulation, myomuscle-genic initiating autoregulation. So it initiates its regulation of its own muscle. And so if there were uncontrolled increase in blood pressure through an arteriole, so let's say for some reason something happens and you have this huge surge in pressure then in one particular arteriole, then you would think, right, by what we've learned, that flow just increases a lot and you increase the flow dramatically. But that's not always a good idea, to increase so much flow to a region. So when smooth muscle fibers stretch in the arteriole due to pressure, when they stretch excessively due to pressure, they will, as a result of that, constrict. It's kind of like in the heart. Remember when there was more filling in the heart, this Frank Starling law, and then it would contract more forcefully. It's very similar to that. So here you have a surge in pressure. What it'll do is it'll constrict, vasoconstricting there. So interesting what the mechanism is. It does it on its own. It's not coming from the brain or from a hormone or a neurotransmitter. So what happens is that the stretch opens mechanically gated calcium channels. So remember, mechanically gated sodium channels also. You stretch it, and if you're able to stretch that, then you are going to open those calcium channels, calcium flows in, and you're going to have contraction of that muscle. So it's a very cool localized mechanism. Paracrines now can do that systemically. They can alter vascular smooth muscle contraction, and that's different. Here you have some sort of paracrine that's traveling in the blood and it's going to a different part of the body and it's going to cause that change, causing vasoconstriction or vasodilation. So not isolated to one region. In your text, table 5.2 has 14 different chemicals that mediate vasoconstriction and vasodilation. So that's a lot. And if you're interested, you should look at those. But really, at the level of, for this class, we're only going to look at 2. Okay, and one of them is the effects of low oxygen and high carbon dioxide. It makes sense that if oxygen is low and carbon dioxide is high, then that's going to dilate arterioles because we want greater flow and we want to bring more oxygen to that area. So an example is increased activity. You're out exercising. You need more oxygen because you now have muscles that want to make more ATP. Then it's going to result in a vasodilation just because of those sensors that you have for oxygen and for carbon dioxide. Okay, that's called active hyperemia. So you're active, you're working those muscles, you need ATP. In order to make ATP, you need oxygen. You're going to vasodilate and bring more oxygen, more blood, and in other words, more oxygen to that area. The second one I want to look at is nitric oxide. So, NO, this is going to trigger vasodilation. When in the world would you release that? Well, when carbon dioxide and hydrogen ions accumulate in the interstitial fluid. So you have too much carbon dioxide, you have hydrogen ions which are bringing the pH down, so pH difference in CO2, then the endothelial cells will start producing this nitric oxide. And vasodilation is going to happen. And the result of that is you're going to increase flow. Vasodilate, increase flow, bring more oxygen to the area. So irrigate it. That's all we're going to say about those. And again, if you'd like to see the others that are causing vasoconstriction and vasodilation, please look at that table in the text. We're going to talk very little about the lymphatic system, but I did want to say a few words about it. It is listed in your reading. These are the vessels that also interact with the cardiovascular because they are the ones that are picking up that excess fluid that is in that extracellular area. Interstitial fluid doesn't sit there forever. It's going to be picked up by the lymphatic system. Very important drainage system of the body. So written out for you, it's to pick up fluids and proteins that have leaked out of capillaries. We have to have the mop-up crew, and this is what this is, the lymphatic system, and it's going to bring all that fluid and return it back into the blood. It's going to put it into the right atrium, and so that extra watery fluid ends up as part of the blood again. How does that work? Here's some pictures of that. So if you have a capillary bed, and this is the arterial end, and the other end would be the venular end, immersed there among those are these lymphatic vessels. So you want to place them then anatomically in a place where they'll be able to pick up that excess fluid. Because remember, this capillary bed is going to leak. And if it's going to leak, we're going to have to pick up some of that excess fluid. But notice where the pickup is happening. About 90% of the fluid is returned to the blood already in circulation because remember net filtration and net absorption? So it exits, but then much of it will go right back into circulation again and it's not lost. That's about 90% of it. But what about the other 10% that stays floating around here? That will be taken up by the lymphatic capillaries, 10%. And moved through these capillaries. And this is a map of all these vessels of the lymphatic system. So imagine it moving then here, coming up and eventually dumping here into the spina vena cava for it to come back into the right atrium. On the right of this diagram, there are pictures of the lymph ducts and also of the lymph nodes. So this is, remember, also part of your immune system. And in the immune system, you've got lots of white blood cells that you can disperse. And these are, just think of them as immune regions. You know that you have the enlargement of some of these lymph nodes when you're ill because you have cells migrating to the area, white blood cells. But for now, what I want to stress is that these vessels are picking up excess fluid and returning it to circulation. What happens if they don't? So edema is the accumulation of that fluid in the tissues. And here on the right side, this is a test for edema. You can press, in this case, on this individual's leg, and then you stop pressing and this stays behind. You kind of push that water around and then you can see your fingerprint left behind. That is the common test for edema. So this is definitely a case in this diagram here. I'm saying the fluid gets trapped in the tissues when these lymphatics are blocked. Abdominal edema also, this is an example of a child who's malnourished and has the big belly. What's happening there? Well, in malnutrition, and these plasma proteins that are made in the liver are diminished or in low number because they're so malnourished that they're unable to make some of these plasma proteins. So if you're unable to make those plasma proteins, then the osmotic pressure within the blood is now changed. Remember we said that it had osmotic draw into the blood because of those proteins. Well, what if those proteins were not there? Then all of a sudden more fluid is going to leak out of the circulatory system and end up in that interstitial fluid, and it can be seen in the abdomen here of some of these malnourished individuals. I'm saying that in this slide that the osmotic pressure is actually reversed. Instead of being greater and drawing fluid in, it's actually drawing fluid out of those capillary beds. And that's the last slide of this section of Blood Flow Part 2 of Module 11. Thanks for listening. Cut it off now. Thanks.